All right. Well, thank you everyone who is joining. So it is my pleasure to introduce Dr. Vincenza Colonna as our speaker today. Hopefully she will be visiting us here in Memphis during the summer of 2022. So I think this is really a great opportunity for an introduction to her research and expertise. And there's already quite a few people here who are collaborating with her. So this will be a great opportunity to welcome her. So Dr. Colonna is currently a researcher at the Institute of Genetics and Biophysics of the Italian National Research Council. Previously, she trained in evolutionary biology and obtained her bachelor's and graduate degrees at the University of Napoli Federico II, which I learned is the oldest non-sectarian university in the world. That's right. Thanks, I didn't know that. And the National Research Council in Italy. Dr. Colonna also served as a lecturer in genetics and biological databases and completed her postdoctoral work at the University of Ferrara and at the Wellcome Trust Sanger Institute. Dr. Colonna is a population geneticist and bioinformatics expert with a focus on human populations and data. Currently, projects in her lab include the genomics of recurrent pregnancy loss and the development of methods to study population genetics using pangenomics and pangenomic methods and models. And this year, Dr. Colonna also received a local Rotary Club Award for her team to work on identifying genetic factors that play a role in CAG repeat instability in Huntington's disease. Over the course of her career, Dr. Colonna has led research projects on the analysis of human genomic sequence variants, population genetics analysis to infer demography and signatures of natural selection in human genomes, evolutionary medicine, and the genetics of rare genome variants. Many of these projects have been and are massive in scope and in impact, such as the 1,000 Genomes projects, and she has authored many high impact publications which have been cited over 18,000 times, according to Google Scholar. And finally, I want to mention that Dr. Colonna is also a highly qualified teacher and trainer for statistical analysis and research computing, and is the founder of the Open Bioinformatics Laboratory, which is a nonprofit project designed to educate and provide services in bioinformatics to students and researchers. So Dr. Colonna, welcome, and thank you for agreeing to join us during your maternity leave.
Thank you, Megan. It was a great presentation. I was like, thank you so much. I'm just going to share my screen and start the talk. Okay, I think everybody can see it now. Can you see my screen?
Yep, looks great.
Okay, so I should probably. Do you also see people on my screen? I should probably do this. Okay. I have the Zoom bar on the side, so I don't know if you see it. Okay, anyway, it's fine. So, so the title of my talk reflects the projects that I've been working on in the past few years. But before talking about the project, I also would like to introduce you to the reason why I got interested in this particular topics. And it all started with my contribution to the 1,000 Genome projects during my postdoc at the Sanger Institute, and that's an image from the last paper of the 1,000 Genome published in 2015, where there's the number of differences for every genome of the over 2,000 individuals analyzing the genome in the project. These are the difference compared to the reference sequence, and every cross here is an individual, and on the y-axis is the number of sites which are variable compared to the reference genome. And individuals are subdivided by continental population, and within continent by let's say nationwide populations. So what you see here is that on average, a person differ at 4.3 million sites from the reference, but that's the average. And then you have European people of European descent, which are very similar to the reference sequence, and then people from America and Africa, which are are the most divergent from the reference sequence. And this is actually in part reflect something, a true phenomenon, that is like that African populations are the oldest, and so they have accumulated more genetic variations. And as for the American population, most of these are admixed with Africans. So this explains the pattern, but also the major thing is that this lack of representation in the reference genome of the genetic variability of African and American populations. And so this is like something to keep in mind that we're going to discuss in a few slides. This is a major limitation of using a reference genome for understanding genetic variability. So within the custom genome, I was interested in studying natural selection in human, and in particular, I did studies on negative selection and positive selection. And just to remind to this audience that what negative and positive selection are, when we talk about negative selection, we talk about selection against extreme value of a specific phenotype or trait, whereas when we talk about directional or positive selection, we talk about selection against a particular range or in favor of another particular range, usually like extreme phenotypes. So talking about, I'll tell you like two stories. One is about positive selection. The other one is about negative selection. And the way we studied positive selection in the Towson genome was through the analysis of the allele frequency of derived allele. And just to make sure that everybody understands what the derived allele is. So this is a phylogenetic tree at a specific site in the genome. And those are our closest relative. And those are the time that separates each branch. And so imagine at the particular position, the ancestral genome had a C, and this was inherited by most of the lineages. But in human, a new mutation appeared, and this should have happened quite recently, of course, in evolutionary timescale. And so this is called the derived allele, and this is specific of human lineages. And the assumption is that the derived allele are the most likely to be relevant for human evolution because they're specific to human. And also the other assumption is that high frequencies of derived alleles may not occur randomly. And so the way we studied like positive selection was comparing the derived allele frequencies among pairs of population. So this is the statistics, the delta DAF, which is very simple. It's just the absolute value of the difference in allele frequencies. So I produced many of these like Manhattan plot where every dot is the delta DAF at the specific site, not the delta DAF itself, but just like the minus log 10 of the p-value of ranking that particular site at genome-wide level. And to make a long story short, like the higher is this value, the more there's difference between a pair of population. And in this case, this is a comparison of African versus Europeans, and so at continental level. And this is an example of a comparison within a continent of the people from UK and people from Italy. And you see two examples of genes which are known to be under selection, the dark gene and then the gene of the lactase. And so beside these known genes, those known example of natural selection, we discover a wide set of other sites which could be considered, which could be potentially interesting. And in fact, we demonstrated that many of these sites are found in genes or in promoters of genes, which are a central node in a network of genes. Like this is a network of all possible genes and the human genes and their interaction. And the largest is like every circle is a gene, and the largest is the gene, the highest is the number of connections that the gene has. And so potentially the more relevant is for metabolism and for life. And so what you see in red is genes which have high D in promoters, in blue genes that have high D, which are missense sites. But that's another story. What I want to say here is that when there's natural selection, what happens is that the study is made at level of the haplotype because of the a phenomenon which is known as selective sweep, for which the selected variants, imagine those are all chromosomes and those are all genetic variants, and the blue one is under selection, and it happened to be in linkage with these other variants. And so what happened is that if this haplotype of the person carrying this haplotype is favored, then after a while, this haplotype become prevalent in the population. And then after a certain time, you get eroded by recombination, but for a certain period of time, this is prevalent. And so the whole study of selection is done at haplotype level. And in fact, we also demonstrated that these sites are under positive selection using two statistics, the integrated haplotype score and the cross-population extended haplotype homozygous is a very long name, but the point is that like those are the sites which have a high derived allele frequency in African, in Asian, and in Europeans. And this is the IHS for the sites, and the gray are the IHS and like SPHH for sites which are matched for allele frequency. And what you see here is that you know, the sole proof of positive selection is based on information of the haplotype. But we know, and I don't know if you can see my my pointer here, because I don't have a red pointer.
Yes.
Yeah. Okay. Good. Cool. Yeah. And so we know that the natural selection is started based on information content of haplotypes, but we also know that genomic haplotypes are not fully resolved. And this is a recent paper from the telomere to telomere consortium that shows the actual telomere to telomere assembly of a genome of this particular cell line, which is an haploid cell line. And I just was magnified area of this beautiful image that shows for each chromosomes the gaps in black existing on the reference, the most up-to-date reference, which is the H38, and here is the amount of genome in megabase pairs that has been added to the information of the reference by the telomere to telomere assembly of this particular genome. And you see that in particular on chromosome 9, this assembly added a very large region that contains many novel genes. So you know, that's, you know, and I think I just want to stress that it is important to consider haplotype with this particular case that we followed up from the Heidi project. This was a SNP in a gene which is involved, which is expressed in the skin. And this particular SNP has a high derived allele frequency in population out of Africa. And so we thought that, you know, because like we got a signal, this is the delta dove, and the bluish like dots are comparison of African versus non-African populations. And so we got a very strong signal here saying that the allele frequency of the derived allele at this size was particularly high in population out of Africa. And when we measured the XPHH, again, each dot here is a window, and on the y-axis is the value of the XPHH for that particular window in pairs of population. And whatever is within the dashed line is not relevant, but whatever is outside the dashed line implies signal of positive selection. And what we see here is that this is the variant of interest, but in fact, we got a signal downstream on an haplotype that is in a strong linkage disequilibrium with the variant of interest. And so this is just to say that when we try to functionally validate this variant, we failed. Oh, sorry, I just want to say this gene is relevant because it causes a range of phenotypes that could be severe and leading to death. but also can be tolerated. So, you know, for all this reason, we thought it could be a nice case to follow up. But I would say, going back, we were probably following the wrong variant because we were not aware of the whole haplotype content. And so this is like an experiment in which we evaluated the gene expression in relationship to the 3 different genotypes. And we did it on mRNA from pullet A roots because that's a proxy for skin. We might have used the wrong tissue or whatever, but like it just like we didn't see any particular difference here. Okay, so that's really not relevant. So I just want to say that the study, I mean, and when I was studying positive selection, I kind of become aware that there are limitations. And those limitations are mainly the fact that we have the genetic variability. We, as we discover it, is biased by the reference, and I'll explain this in a second. And also that we don't have full awareness of the information of the haplotype. So a way to deal with, especially with the first limitation, is the use of pangenomes. What is a pangenome? So in the usual approach, which we now call genomic, the reference sequences compare every sequence that is produced is compared with the reference sequence. And when a novel sequence is produced, this again, this is compared against the reference sequences, and differences are detected only against this particular sequence. With the pangenomic approach, every sequence is compared against each other. And when a new sequence is produced, then the differences are evaluated in relationship to every possible sequence. And so that increases the number of variant and the type and the quality of the genetic variation that we can discover. So the way we build the pangenome is through a few steps, and we use a software that has been developed also at UTHSC. And so the first step is to align the sequence against each other. So let's say for a specific sample, we have several sequence information and everything goes aligned against each other. And then, so this produces an alignment graph, and then the information of the alignment graph is organized through a variation graph, through a process of like inducing a variation graph, in which some information is condensed, like and the graph is produced where there are nodes that represent segments of a certain part of the genome, which are unique, and edges that connect the segments. And in this way, you have all possible, you have a complete representation of all possible paths that could happen in a genome in relationship to segments of genetic variation. So this is in theory. In practice, it's kind of like messy things that get produced, and then it goes through several steps of sorting and rearranging. But at the end, what we got is in this particular representation, you have a sets of genome which are aligned to each other, and the genetic variability is displayed as regions that are shared or not shared. For example, like in this case, this is an insertion that is specific to these few genomes, like every line is a different genome, and this insertion is present only in a few genomes and not in other, and so forth. So this is mostly the work of Flavia, which is a PhD student at UTHSC. She will start soon, like in January. So we were working on producing the pangenome of BXT mice. And I don't have to talk about BXT mice to this audience because you might be familiar with that. But just like, and I hope I didn't say wrong, these are mice which are obtained by several generations of inbreeding, starting from two parental strains, which are kingenic and completely different. And what we did, we had available sequence data of 10x linked breeds and to some extent also a few PacBio and Oxford Nanopore data, but only for the parental strains. And so Flavia used this data to produce the pangenome. And as I said, the main data was like sequence information from the 10X. And then we also, this data was like assembled, and then the peak, and then the contigs were used to produce the pangenome. So when it goes to pangenomes, the longer the better. And so you ideally want to have very long sequence information. The peak of the contig produced by the 10X was at 31 kilobase pair. It's not exciting, but it was a good start. And then somehow Flavia is trying to integrate also data, nanopore data of parental strains, which have contig length slightly higher at 80 kilobase pairs. And also you can use the supernova data to correct the nanopore reads, because the nanopore reads are not entirely and not highly accurate. But those are technical stuff. This is the beautiful thing that Flavia produces. This is the pangenome of chromosome 19. And the way you read these, like every line is a strain. She managed to assemble from 148 strains. And so there are 148 lines here corresponding to each one to a strain. And region overlapping are region of a region which are similar, whereas like gaps are specific deletions or insertion. And this is the region, excuse me, this is the region of the centromere, and this is the region of the telomere, and the alignment in this region is really poor compared to the rest of the chromosome. So it took a while to produce the pangenome, because the software for producing the pangenome was written at the time at which Flavia was actually using it. So she had to to really be she was at the cutting edge of the pangenome for a while. And so this is the last version that we produced, and as you see, it's got a lot of like ends in the pangenome. This is the content per nucleotide, and there's like 50% is ends, and this is due to the fact that we realized after that the 10X linked reads data are not probably the ideal data for building a pangenome, but still it was relevant that she managed to assemble one. And again, as I said, there are a few things, like this is another representation, and this is an extract from this gene, so this is another representation where segments nodes are represented as segments, and those are the edges connecting nodes. And that means that if I go along this sequence, which is just a zoom of this area here, so if I go along the sequence, I have like a region of one base pair was shared by all the 147 strains, and then this here is a SNP, but this SNP is present in like this base pair. This particular base is present in 147 strain, whereas one strain has a different base here, and so forth. And this is an insertion of one base pair that is present in 2 mice. And again, this area, I mean, this is another way of representing the graph. So the length of the noses in base pair, and then the depth basically corresponds to the allele frequency of the polymorphic site. As I said, the space for improvement, this is an example where there's a stretch of like 5,000 ends, but this is found only in one individual. So, these are things that needs to be smoothed. And again, Flavia was really excited. She managed to map a sign in this region of B1, like by comparing the sequence of the stretch by using the sequence of the stretch to search for this database, like she managed to find that is assigned. So anyway, so the pangenomic call set it turned out to be enriched for complex variants, and this is exactly what we wanted to demonstrate. So even with this data, which is not ideal for pangenomic, we managed to discover a range of variants that are not present in the genomic assembly. And when I say this, so this first bar is the variant call set that we obtained from the pangenome. And here for comparison, this is the variant call set obtained by traditional genomic way, starting from PacBio data, and this is from 10x data. And what we see is that in the pangenomic call sets, we have a slightly higher number of variants which are complex, which are indicated here by this like purple color. So I can probably skip this. Just like briefly, I just want to say that for SNPs, we obtain like values of precision which are comparable to what is found in genomic comparison. So, this is quite good call set. So what we use the pangenome for, like what's the usage, what's the reason why we're so interested in producing pangenomes? Because we can get a lot of information from there. And in particular, this is a nice example of a specific insertion in the Zeta PF91 gene. And so what you see here, this is again a representation of the pangenome, and what is green is specific of DB-ATJ and 70 BXT mice of the 140 that we analyzed. And whereas in orange, this is not found in DB-ATJ this is typical of another 77 BXT strains that we analyzed. What happened here is that this insertion, which is specific of DB-ATJ, was inherited by half of the inbred mice, and which is exactly what you expect. And so in practice, this is to demonstrate that the pangenome can be useful to discover like strain-specific genetic variability. So this is a project that Flavia just started while she was doing, in fact, her master thesis. She's very brilliant. So the future perspective for this project is to, first of all, focus on constructing a better pangenome. And this depends on both data and tools. And for the data part, we're already planning to sequence BXT mice using Oxford Nanopore technology, like long read technology. And also we have available through collaboration some data, some PacBio AFI data of other mice strains. So our first focus will be on building a better pangenome. This depends also on tools, and this is in fact in continuous development and updates. So we also depend on that. And once we have the better pangenome, we can focus more on biological and evolutionary aspect related to mouse genetics. So this was like for the first part. And then I'll set now a different kind of different part of the talk about different topic, but always related to natural selection. So as I said, my interest was in studying human selection in humans, and we discussed about positive selection. Now we're going to talk about negative selection. And again, let's go back to the 1000 genome where it all started. So the assumption for negative selection is that higher fraction of rare variants is indicative of selection constraint. And just to explain this quite complex slide, so these are two examples. This is a region of the genome where there's prevalence of common variants. And this is a region of the genome where there's prevalence of rare variants. And the idea is that this region is probably under selection because it is constrained and it can't actually change. And if something happened, this stays rare because it's not favored by evolution. And so and so the statistic for studying negative selection in the 1,000 genome was this fraction of rare variants, and where for rare we used the frequency of 5%, but like, I mean, the threshold for defining a rare variance was 5%, but of course you can play a lot with this threshold. And the way we did it in the 1,000 genome was to, we were interested especially in non-coding and the non-coding part of the genome. And so we subdivided the non-coding genome in functional categories. And there were 677 functional categories. You see enhancer, and then transcription factor binding site, DNA hypersensitive region, and pseudogenes, and so on. And then we measured the RDAF, like the fraction of rare variants in each of these regions. And what you see here, these are the top 24 categories. So on the y-axis is the fraction of rare variant. And this dashed line in orange is the average for coding region in the genome, which are known to be, well known to be under negative selection. So we used as reference. And this is the genomic average. So some of the categories that we identified, they have a value of RDAF, which is compatible with the one of the coding region. And so we define those are ultra-sensitive regions. And then we define the second categories, like the sensitive region as the number of categories whose genomic content was comparable in size as the exomes. And again, I want to stress that here the coding genome is considered as reference for negative selection because it's well known to be under negative selection. So again, this is an interesting way of defining negative selection, but it was based on alive, I say, like healthy individuals, and it doesn't consider cases of strong negative selection. And so this was like, let's keep in mind this, and I'll tell you after how I think we can deal with this problem. Again, so we demonstrated that the sensitive and the ultra-sensitive region have a fraction of rare SNPs, which is comparable to a coding average. And like, the only category which was like higher is the missense variation, which are known to be under strong negative selection. And the ultra-sensitive region contains several categories of transcription factor binding sites. And in particular here, we highlight 3, which are particularly rich of rare variants. So this is just to say, we use this information to incorporate it in a pipeline for prioritizing genetic variants in non-coding region of the genome. And this is the pipeline. It starts from like variants found in the 1,000 genome, and then it integrates functional annotation, and then the information of being in sensitive or ultra-sensitive region. And then if so, then it is checked if they this RAP motif in known like a transcription factor binding site, for example. And then we also evaluate how central they are in the network of genes. And we applied this pipeline to cancer. And so we were also including information on cancer. But again, so this was just to complete the story of the negative selection in the 1,000 genome. But as I said, that was interesting, but I still think that one limitation was the lack of model study for negative selection in human, because like all the individuals we analyzed, they were alive. A way to deal with that, I think, is the study of natural negative selection in individuals that do not actually live. And so to do that, you need to look at early embryonic development in human. And we did that by looking at 4 particular stages of human embryonic development. The first 3, so I'm just checking the time here. Yeah, so the first 3 are visible in IVF treatments. And that's the maturation of the oocyte, the fertilization, the rate of fertilization. And we know that like fertilization fails for 5–10% of IVF patients. And also the failure of embryos to implant, and this happened in 50% of all IVF treatments. And then a second set of like case study that I found interesting is the embryos, and in particular the miscarriage, like the recurrent abortion. And so that's miscarriages happen in 10–15% of all pregnancies, and this is a clear sign of negative selection. I mean, I would say there might be many causes for every of this phenotype to happen, and not all are due to a specific genomic causes. And I would say, in fact, most is also due to in this particular case, to technical limitation of the IVF. And in this case, it could be due to incompatibility of mother fetuses, or just infection, or any other phenotype. But for sure, those cases are enriched of cases in which there might be a genomic reason for life to fail. And so this is an interesting case of negative selection. So we, as a pilot, to make a pilot study, we consider 28 mothers that belonging to any of these phenotypes for which we had exome sequencing, and 10 miscarried embryos for which that we collected and for which we produced the whole genome sequencing. And those numbers are going up. We already have another like 15 miscarried embryos ready to be sequenced, but you know, I'm happy we managed to do the pilot project already with 10. So Sylvia, this is the work of Sylvia. She's a brilliant PhD student. She is about to finish her PhD. Sylvia set up a pipeline for prioritizing genetic variants, because you know, of course, the question was which genetic variants are relevant for these phenotypes, and basically the first step is a filtering step where the variants are filtered based on their impact and based on the frequency in reference population and based on the functional effect. And we're interested in variants which have a high or moderate impact and a rare, or, you know, like, again, we can play a little bit on this number, but rare in the population, but not so rare. And then also they should be highly damaging and in gene which are intolerant to loss of function, or they should be in genes which are relevant for embryonic development. And there's no time to go into details of the pipeline. Just want to say, you know, this filtering step is followed by another set of filter that check for technical artifact, and especially we have a filter that check if in a control population, if the filtering strategies that we are applying, it's just producing results by chance. And I'll explain a little bit better this part. So we have a control population, and we usually use the HEDP samples. And so we sampled 100 times a small set of individuals, and we apply the same filter that we apply. And we do this like 100 times, and then we have a set of genes and variants. And so we just say these are the genes that would be filtered by chance by our filtering criteria. And so we use this information to clean up the results. So as I said, this work was done in collaboration with Genomics, which is a company that does IVF, and this is Matteo, which was doing the work with Sylvia. So for the first set of cases we had, we selected those women that had a very low oocyte maturation rate. So these are our cases, and this is the average rate, and in blue is the average rate per class of years, because the age is also important. And so the dot is like the average of the up to 10 embryos up to 10 eggs, sorry, for the same women. And so they all have a rate of maturation which is way below the average. And that's the same for rate of fertilization and the rate of implantation. So we selected all these cases. We did exome sequencing, and we discovered a number of genetic variants in 900 genetic variants in 800 genes, and 20% were not in candidate genes. So we were able to discover also novel gene. The way we read these results, this seems very complex, but I'll go to the next slide. Sorry, yeah, I'll go to the next slide, it's easier. So for every individual, these are the gene, and those are the variant prioritized in that specific individuals. And the color indicate if the variant is heterozygous or homozygous, and if it's moderate or high impact. And so you have for every individual a list of variants in genes, and some are genes which are known, or they just moved, but the arrow should be on PADI6, and this other arrow should be on TRP13, which are genes which are well known to be involved in this sort of phenotypes. Overall, the variants there are a few variants with high effects, and the vast majority is with moderate effect and mostly missense variants. And just going back one slide, the genes that we identified are involved in DNA repair and mitochondrial process, which again are consistent with processes that are deregulated in early embryonic arrest. I just want to check the time. Okay, so almost finished. So for these particular cases, we were able to do some sort of validation in a data set of single cell RNA sequencing from intragranulosa cells and M2 oocyte. So this is the representation of the oocyte in M2 phase, and this is just a graph. So in this paper, they produced data, single cell RNA sequence data of this RNA. And we asked the question if the genes that we prioritized were among the highly expressed in oocyte. And the answer is yes. So in blue are the genes prioritized by our pipeline in our cases. And in gray, we applied the same pipeline to random controls. And both for the intragranulosa cells and the M2 oocyte, the odds of the genes to occur when they contain variants of high impact are significantly higher compared to random control. And more in general, like we can say that like about 50% of the genes prioritized with high impact, they are in between the first 5,000, like ranked in the very first highly expressed genes in intragranulosa and M2 oocyte. So for the embryonic development, we started from 46 embryos, and we first of all selected only euploid embryos because we most, let's say 60% of the embryos, has aneuploidies, and mostly trisomies or triploidies or monosomy and so on. And so we performed like a set of technique like qF-PCR, RACGH, and shallow sequencing to exclude all the aneuploid embryos, and we concentrate on the euploid embryos. And for this, we produce whole genome sequencing. And again, we applied the pipeline for variant prioritization. Here, every graph here is one embryo, and there's a number of genes which are prioritized for each embryo. And sometimes the same gene has more than one variant. Overall, the variants are, there are, again, few variants with high impact and many variants with moderate impact. And we're struggling a little bit to validate. In fact, like the paper is almost, I don't have to say, but the paper is almost accepted. But we were struggling with the reviewer for validation because it's a very difficult stage to validate. But one nice thing is that among all the genes that we found, the TLA4 was shared by more than one embryo, and we would not expect this in such small sample size. But in fact, we found it. I think in 4 out of 10 embryos. And what happens is that this gene has been found, like showed up in one GWAS for sporadic and recurrent miscarriages. So this was done on mothers. So let's keep in mind, it's not in the embryo, this is in the mothers. But what they found in this, in this paper, is that they found a signal in a region that physically interact with TLA4. We also know that TLA4 is implicated in 2 pathways, the Notch and the Wnt pathway, which are important in the embryonic development. And the Notch pathway basically repress the pluripotensive genes, and the Wnt pathway is implicated in the development of the trophoblast, which is the tissue that develop outside the embryo that connect the embryo to the mother. So future perspective for this project, we use this pilot data to get funding for a project that just was funded. I just got noticed, I was notified about funding like last week. And so in this larger project, we will definitely extend the collection of samples to 3 years and also to 4 more centers in Italy, because we were collecting only from one hospital. And also we will extend the phenotype to idiopathic infertility and recurrent implantation failure. So we will have 2 more categories. And what is more interesting and thrilling for me is that we will finally be able to produce long-read data, and also to apply strand sequencing to identify balanced translocations. So in conclusions, I just wanted to give you an idea of also what's the vision, as the conclusion and vision, what's my idea for this project which is going on. And it all started from my interest in natural selection and limitations that I saw in the past years when I was working as a postdoc in projects that were of large impact, but still, I would say, not. Just obsolete now. If you think about the Tyson genome and the impact of the project, which was amazing, but the idea of the quality of the data has improved so much in 5, 10, not even 10 years, that now that project seems obsolete. And so I would say, considering all this limitation, I think that a way to continue in the future is to definitely use pangenomic approaches and better sequence technology like the 3rd generation sequences. And again, and in particular for the study of selection, I believe that phenotypes of early human development could be also an important, a key factor to keep in mind. And with that, I'd like to thank my research group, which is 3 PhD students at the moment, but they're amazing, and just want to say, thank you to them. And we are more than a lab. We're kind of a small family, and we like to work together. And also, I'd love to acknowledge collaborations at University of Tennessee and with Genomics and the University of Ferrara, where the first batch of embryo was collected, and the Monash University in Malaysia, where we are going to collect samples of consanguineous, of recurrent, abortion from consanguineous families. And that's it. Should stop sharing.
Great. That was very exciting. And I guess we're open up to questions. And if no one is willing to start out the questions, I guess I have a couple, a question thought. So I do hear a lot about the pangenome, and I guess it does worry me a little bit. I mean, do you think we're going to have to abandon all of the existing short read and linked read data that we have and completely generate new data? Because it has a lot of implications for, you know.
No, I know.
So maybe
yeah, I know. Yeah, I can comment on that. No, I know there's been so much effort. I definitely think that like third generation sequencing and long read are the future, and that's where we're going. And it's true, the cost, I mean, we were expecting a reduction in the cost that hasn't happened yet, but I'm sure it's going to happen. That was like, you know, the first genome produced with Illumina, they were so expensive. And then you just like, you know, you can sequence the genome now for something like $300, something like that. So definitely this is the future. It doesn't mean we have to abandon all the old data. And in fact, I think, you know, there would be, there's, you know, we should also get interested in studying how can we make, you know, the best out of short reads. You know, I'm really trying hard to say that they're okay, but I can't. I just think the future is in long reads. But it's true, you know, we should find a way to use the data that have been already produced. It's like, you know, it's like the old SNP chip when the short reads came out. And, you know, but they're still useful for something. Even now, SNP chips are useful for many. So I just think, you know, I don't think we have lost something. I think we have, like short reads are important for the amount of accuracy that they produce. And so I think they still be used for validation eventually or for integration with long read data.
Yeah, I mean, maybe more for genotyping, I guess.
Yeah, for genotyping, exactly, yes.
Any other questions? Rob is unusually silent. Yeah, so I was also thinking, I think that's great. Rob had made a comment, and yes, the figures are really cool for the BXD pangenome development. It's nice to see. It's nice to see that project moving forward. So I guess then, I mean, potentially, you know, if you have populations that have been around for a while and not nearly like hundreds of years, but I guess, is it feasible to look at natural selection in laboratory mouse populations as some type of response to actually, you know, being in a lab?
No, I think it's exciting because like well, I think it's going to be easier to look at artificial. I mean, like the laboratory mice that have been strongly selected for many things, and so I think it's going to be even easier to look at selection in mice like compared to human. I would say human are probably the worst species for studying natural selection because they have such long reproductive cycle. You know, it takes a long time to get a lot of generation, whereas like the shorter is the generation time, the more suitable is the species for the study of natural selection. And that's one thing. And second thing is the selective pressure. Like you can't just like breed and apply pressure to humans like as much as you want, but you can do it with mice. So that's definitely, and this particular resource, the recombinant inbred mice, they are even better because of the information content on you know, the linkage map that you can have. It's just like impressive. And so, and plus you can replicate, and also you can, I mean, this is an ideal data set because they have a phenotypic data and you can replicate the phenotype and the genotype as many times as you want because you basically have the same mice and you can reproduce the same mice like many times. So to me, this is an incredible good set for studying natural selection.
Yeah, and I don't know if you were going to comment more, David, but I did notice, so the example that you gave of the deletion, it pretty much looked like it had segregated like equally in the BXD population, but I know of some variants that maybe are a little bit more recent, but still like not as many of the BXDs picked up that variant. So I mean, I don't know exactly.
But maybe
that's
exactly. yeah, that's the point. Like, this was the case like of like 50% segregation, which is what you expect. And what will be interesting is to look at those which are not 50%, but for some reason, they don't get inherited as 50%. And so what's the reason behind that? Like, are they deleterious or they're inherited by 90%, let's say. So are they favorable and so on? So that's exactly the way in which, should progress.
The thing I was going to comment on was with long reads in the pangenomes, we've had problems with like regions that look like they're heterozygous, that it could be sort of a duplication that's very similar and things like that. Whereas with the pangenome long read, because would be really interesting if you've got regions that have stayed heterozygous because being fixed in one direction or the other is deleterious. And that's something we can't get with short reads.
That's right.
Yeah, I definitely can appreciate the power of the pangenomes to find those complicated structural variants that are missing.
Yeah. So the pangenome is only the first step, as I said, and then once we have, in this here, like the whole effort was to build the pangenome. It takes so long, because we were actually in the stage in which the software was literally written. So Flavia was like really at the cutting edge, bleeding edge, Rob says. But now we can concentrate more on biology, and I'm just look forward to do that.
Yeah, my cool idea in the future would be that sort of all sequencing in the world was in one pangenome. So you just go to Geo or whatever, and everything was just in one pangenome. So you'd have every single strain that's ever been sequenced by RNA-seq, DNA-seq, methylation, and you'd have its chain through the genome for each of them.
That's the idea. That's the hope, David. Good luck with with manipulating that. Yeah. Yes.
Yeah. I also really enjoyed the research that you're doing with reproduction. That's really sort of like an interesting way to think about natural selection. And I guess you can correct me if I'm wrong, but I guess the premise is that maybe you're looking at a population where they already have genetic variants that influence reproduction. Is that?
The idea is that some of these failure in the early embryonic development might be due to genetic causes, and of course there's a slide I didn't show. There are several causes, might be environmental causes, might be any other cause, but for sure, this is the best set of samples for studying negative selection, because they'll be enriched in cases in which the genomic component is the actual cause of negative selection. And so I'm aware that you need large number to do that, because 10 embryos is not much, but that's a start. We got funded, that's great. But eventually, I'm sure when we reach a size of the sample which is reasonable, we'll be able to pick cases where the actual reason for miscarriages was some genomic reason.
And these would be mostly inherited, because I imagine there are also de novo that might also, so you could immediately see.
Exactly. So the expectation is that for we're selecting recurrent miscarriages, because we think that if it's a de novo mutation, then it's probably a sporadic case of miscarriages, because it's de novo and it's not going to happen again in the next in the next pregnancy. But for recurrent miscarriages, then you can actually reason that the genetic defect is in the parents, in the mother and in the father, and so those would be again enriched for cases in which the genomic, the genome is the cause.
And I think I saw that there was a pretty big variation in age, maternal age there.
Yes. Yeah, that's right. Because there's you mean in the rate of failure?
In the IVF So in the cases.
Oh, yeah.
So as a consideration, I guess there might be different.
Yeah, of course. Yes, of course. Yeah. I'm sorry. I'm not showing the slides anymore because I wanted to see people.
No, that's fine.
Sorry. Yes, there's a considerable range of ages and differences by age.
All right. Well, yeah.
I believe you mentioned you applied some of this methods to cancer genomics. Did you find any results from that?
Yes, that was like a long time ago, and we were so the way. Sorry, I'm just going to share the slide because it's easier to. What happened? Oh, the things I want to. Didn't put any slide on that because there was like a different story. But yeah, it is. Yes, we were, when applying to cancer, what we were doing, we were taking consideration mutations that were occurring in multiple samples of cancer and prioritizing on that. And when we did that, we were able to predict to some extent, mutation that could be driving the cancer, and we validated that in some like yeast experiments. And I'm sorry, I can't be more accurate because this was really long time ago, and I can't remember exactly all the details, but everything is in the paper. Excuse me, I just can't remember right now. I'm sorry, I didn't answer very well this question.
Oh, no, that's, yeah, no, I mean, and this is great. I think also some of these slides remind me that a lot of people focus solely on, the exome and like missense variants there, but,
oh yeah,
the non-coding portions of the genome that we don't understand as well.
No.
Kind of like, I don't know, could you call them like potential sites of actions for adaptation? Because they're, I don't know, they're not necessarily under the same kind of selection restraints.
No, absolutely, and sorry, absolutely, and the focus on non-coding is like there's a part I didn't present, like Sylvia. So we applied the prioritization pipeline in the coding region because these are the most easily annotated. But like what Sylvia's doing, she is also studying the non-coding part of the genome. She's trying. So the way we want to use this information is we want to determine region under negative selection in a new data set, like some sort of like the work we did in the 1000 genome, but she's doing this in the NOMAD and in the HGDP data sets. And then once we found candidate for negative selection, those are going to be within the prioritization pipeline. So let's take some evidence from the data set we have, and then use this to prioritize in cases, like to prioritize variants in cases of miscarriages or like embryonic failure in general.
Yeah, and that, I mean, I imagine that would also help understand some of the transcriptional and regulatory processes that happen in early development.
Absolutely. Yeah, absolutely.
Well, any last questions? I know, I mean, I hate to take up too much of your time, and I really appreciate you being generous with your time and coming to present your research. Any last questions? One last call. All right, well, thank you, Enza.
Thank you.
I really appreciate it. Can't wait to see you in the summer when the weather's better.
I'll see you soon. See you soon, yeah.
And have a great weekend, everybody.
Thank you, bye, bye-bye.